Good day, friends. You're welcome back to the channel. I do hope you are enjoying the content that we put out. Please don't forget to like our content, share our content, subscribe to our channel, and help us to keep going. We are aiming to reach 1,000 subscribers, so help us to keep pushing this. Today we are going to discuss gram staining. Gram staining is an important concept in microbiology, and the idea being that there are a lot of bacteria. Exist, and for the process of identifying these bacteria, many methods and techniques are being used in the lab. Now, at the root of many of these techniques is a technique that we call gram staining. This gram staining was designed by a scientist called Christian Gram in 1884. And I'm going to run through the procedure for this gram staining. And it's important. It's important because it helps to divide most or all the bacteria that exist into two major groups, either a gram positive group or a gram negative group, a gram positive or a gram negative group. And this is important to know. Now, the process of the gram staining is quite easy once you understand the science, and it's easy to know. You don't need to cram it. It's easy to know, it's easy to understand when you know the science behind it. Now, for the gram staining, the first thing that has to do is you have to heat fix a smear. Now, we know that bacteria, in order to be able to gram stain it, you need to have the sample of the bacteria. You need to put the sample of the bacteria on a glass slide. When you put it on a glass slide, you can put a drop of water on that glass slide and then run it over a flame very quickly, or you can leave it to air dry. So the idea is to create a smear. You now have bacterial cells on that glass slide, and they are dry and ready to go. Now, when this has been done, that's what we call fixation. You prepared a smear and you fixed the smear. The next thing to do is to begin the actual gram staining procedure, and this starts by adding the first stain or the first chemical, which is called crystal violet. It's a primary stain. We call it the primary stain, and it's crystal violet. Now, crystal violet, which is the primary stain, is added on that smear. And when we add crystal violet to the smear, crystal violet is purple, violet in color, and it begins to react with the cell wall of the bacteria. Now, once this is done, the next thing to do is to add iodine. Iodine is what we call a mordant, and we know that's why I said you don't need to cram it. If you notice, iodine and crystal violet, they have almost the same color, purple, or violet. So iodine is the next thing to be added, and iodine is what we call a mordant. It's a mordant because it helps the primary stain, which is crystal violet, to stick even stronger to the walls of the bacteria. And it does this by forming complexes with the crystal violet. So you have what you call a crystal violet iodine complex that is formed within the cell wall that gives the cell wall of the bacteria this is purple color. So that's the second thing that we add. Now, after that has been added, the third thing that is added is what we call the decolorizer. The decolorizer, and the decolorizer could either be acetone or ethanol. Acetone or ethanol, any one of the two of them will do. So this is also run over the smear, and we try to run it very well we run it over the smear. Now, when that has been done, the work of the ethanol or acetone is to decolorize, to try and wash off the primary stain, depending on the kind of bacteria we are dealing with. Now, after the decolorizer has been added, the last and final thing to be added is what we call carbofuchsin or safranin. This is the last chemical that is added for our gram staining. Now, when this chemical is added, the chemical has its own color, and its own color is pink or red. And so you now have a situation where one of two things happens. Is either the bacteria retains the primary stain, meaning that it will remain as purple or violet, or it won't be able to retain the primary stain, and then it will now hold on to the secondary stain, which is pink or red, which is safranin, carbofuchsin. Now, the issue is, how do you understand what is the science behind whether we are going to have it staining red or staining pink? Now, the thing is that for your gram staining, we have to realize that the gram positive bacteria, they do have a thick peptidoglycan cell wall. please note this very well. The gram-positive bacteria have a thick peptidoglycan cell wall, and this thick cell wall is able to retain the primary stain. So the gram-positive bacteria, it stains purple. It keeps the primary stain. The secondary stain is not able to displace the primary stain from that gram-positive bacteria. Now, for the gram-negative bacteria, they have a thin cell membrane, so they are not able to hold on to the primary stain. What do you call it now? The secondary stain, safranin, displaces the primary stain, and so it looks red or pink under the microscope. I hope you understand the science. The science is very important to understand. The gram-positive bacteria have a thick cell membrane, so they are able cell wall, sorry, not cell membrane, cell wall, and so they are able to retain the primary stain. The gram-negative bacteria have a thin cell wall. They cannot retain the primary stain. The secondary stain is able to come and wash away the primary stain. Thank you for listening to this lecture. Make sure you understand gram staining. You can play it a couple of times. Please like our content, share our content, subscribe to our channel, and have a wonderful day. Thank you.